Hi, everyone. I'm Carrie Bearden. I am a member of the Scientific Advisory Board of OneMind and a professor and a scientist at UCLA. And today, it's my great pleasure to have the opportunity to speak with Arpiar Saunders, who is a 2023 Rising Star Award winner from OneMind. So first of all, Arpi, congratulations on this amazing award. You had a really exciting application, so I'm really excited to ask you some questions about your work and find out a little bit more about the science. So Arpi, your research is aimed at understanding how genetic variation controls the function and organization of brain cells. Can you unpack a bit for our audience the role that genetic variation plays in our brain's organization?
Sure, I'm happy to. Thank you for the opportunity and for your time to come and chat with me. It's really a pleasure to join the OneMind community. So genetic variation, I think we should define the terms. We each have 23 chromosomes, and we inherit a copy from our mom and a copy from our dad. And the sequences of DNA at each of those locations, the chromosomes, are different, right? And that's the variation that occurs within our genome. And that variation changes the properties of cells and tissues and organs. And with humans, partly contributes to our individual differences. And so I think it's actually at the very early days of understanding how genomic variation changes the way the brain is organized and how those effects contribute to human disorders and human illnesses, and also just who we are. So I think it's very early days, and part of the challenge of understanding how genetic variation affects the brain is developing new experimental systems to study genetic variants. For example, one strategy could be humanizing mice, so moving mutations from humans into mice. Another strategy is taking skin cells from human patients and turning them into complex culture systems that can be grown into organ. And another strategy is studying non-human primates and kind of getting a sense of what the primate genome is like and how that builds primate brains.
Yeah, it's some of these things really sound like science fiction, probably, but it's amazing just how the technology has advanced in very recent years, and you're really doing some of the the most innovative work in this area. And going back to your work in your lab utilizes a three-pronged approach to experimental design. So invent, explore, and test. So can you tell us a little bit more about each of those techniques and how they're really working together, and then how you came to utilize this approach in your work?
Sure. Yeah, there's definitely a need to develop new methods to build out the experimental playbook for how we study genetic variation. And so our lab does that by developing methods, using them to test hypotheses about how the brain is built, and then validating those hypotheses with more traditional methods. So that's the strategy. And our area of focus is developing technologies that are based on single-cell genomics. And single-cell genomic methods allow us to sample molecules in thousands and thousands of individual cells, and in a bottom-up fashion, build a picture of complex tissue built from the constituent cell populations, the building blocks of that tissue. And it's really important in brain because, if you look at a slab of brain, right, you have neurons and astrocytes and cells of the vasculature and oligodendrocytes that are all jam-packed right next to each other. And you really need to be able to study single cells to identify the component parts.
And then just in terms of some of the techniques that you're using, two of the techniques is molecular barcoding and single cell genomic measurement. So you mentioned the single cell work. Can you tell us a little bit about the molecular barcoding and how you're using that?
Sure, I can give you an example. Molecular barcoding kind of sounds like a fancy term, but it's actually just using small snippets of DNA or RNA to track individual molecules in cells. So it's a handy experimental tool to help us make really accurate measurements of individual molecules. And one example of the way we're using barcoding is in engineering viruses to teach us about the more complex cellular features of the brain. For example, synaptic connectivity relationships, right? So brain cells have really complex anatomy that can stretch, centimeters across the brain and make synapses with different types of cells that are extraordinarily small. And in fact, they're so small that they're challenging to study with a light microscope. And so in order to study the synaptic organization of the brain, we engineer viruses like rabies virus that transit synapses. So they've evolved to hop from cell to cell using synaptic junctions, and we can use molecular barcoding to track those events and build systematic descriptions of brain cell connectivity. So in our case, what we're trying to do is really take viral engineering kind of into overdrive, and by combining viral barcoding and single-cell genomics, be able to generate very quantitative and efficient descriptions of the nervous system.
And this was basically a new technique that you developed, right? The SBARRO.
Yeah. So the technique that I've developed is called SBARRO, which is kind of cheekily named after the airport pizza chain.
Yeah.
It stands for Synaptic Barcode Analysis with a Retrograde Rabies ReadOut. It's kind of a mouthful, so kind of a cheekily name helps the medicine go down.
I like the acronym. Yeah.
Yeah, and I developed it as a postdoc in Steve McCarroll's lab, which is at Harvard Medical School in the genetics department, and part of the Stanley Center, which is at the Broad Institute. So as a postdoc, I kind of laid the foundation for SBARRO and published the proof of concept work that we could track the infection of individual rabies virus particles. So the way the system works is all synaptic circuits have two layers, right? There's a layer of postsynaptic neurons, which are the listening neurons, and each postsynaptic neuron gets inputs from hundreds or thousands of presynaptic partner cells throughout the brain. And we can consider these the singing neurons, right? And each individual postsynaptic neuron kind of gets a chorus of inputs from its presynaptic cells. And so our strategy using SBARRO is to take thousands of instances of these kind of personal choruses that individual neurons get, and to use them to build these quantitative maps of connectivity. So there's many different cell types in the brain. So if you have a certain cell type, that's the listening neuron, it gets a certain type of chorus of inputs. And then each of the input, the singing neurons, itself has its own chorus that it's listening to. So by breaking apart the neural circuits into these two-stage systems, we try to reconstruct a whole from many sums of the parts.
I really like the analogy of the chorus with the neurons. I hadn't heard that before. I like that. Yeah. And so essentially you're using the virus to trace the path from the postsynaptic to presynaptic neurons. Is that?
That's right. So the virus moves in what's called a retrograde fashion. So we engineer the virus so it infects an individual postsynaptic neuron. And in our case, the barcoding introduces a small nucleic acid fingerprint into the viral genome. And so when the virus does its thing, which is clonally replicate its genome, right? So you have one viral genome that then turns into thousands. Those thousands of genomes each have the same barcode fingerprint, and they're packaged into virus particles, which then spread into the presynaptic network of the cells, the singing neurons, right? And then by sampling the RNAs from all of the cells in the analysis, we can use the barcoded RNAs, the fingerprints, to figure out which cells are making synaptic connections on which other. And by sampling all the other RNAs from the individual cells, that teaches us really important information about the nature of the cell. So its molecular type, its molecular state, and importantly, when it comes to genetic variation, right, we can use these kind of very molecularly driven experimental strategies to start to manipulate the brain simultaneously. So we can use molecular manipulations that themselves are trackable through RNA to multiplex or do many experiments in an individual animal at a time.
It's super cool technology. I just have to say, it's really, really exciting. So, and you're part of the team that created DropViz, which is technology that allows researchers to explore the mouse brain through single cell expression. Yeah. So how does this technology work?
Yeah, that's a great question. So when I was a postdoctoral researcher in Steve McCarroll's lab, I collaborated with several of the McCarroll lab members on an atlas scale description of the mouse's brain. And this was an important step because in the McCarroll lab, there was a really breakthrough single-cell RNA sequencing technology that was developed by Evan McCosco called DropSeq. And DropSeq kind of opened the door for us to sample RNAs, not from 1 or 2 or 10 cells, but from 10,000 cells a day. So I was kind of in the mix with this incredible technological breakthrough, and we decided that the most impactful and important thing we could do was simply describe the molecular patterns that we saw in different brain cells across the mouse's brain, which is a genetic model system that many neuroscientists used. And so DropViz is the name of an online analysis portal that allows users to access this single-cell RNA sequencing-based description of molecular diversity in a mouse brain.
Was there anything that you found that really surprised you?
Yeah, I mean, one thing that's I think really fun about the single-cell genomics descriptions of brain is that everyone kind of brings to it their own experience, right? The data sets are so rich and complex that if you have certain expertise in a brain region or, a set of genes or a set of signaling, you can take that view and look at the data and find and discover amazing new things about the systems that you have expertise in. So for me, as a PhD student, I was really focused on subcortical neural circuits through that have been implicated in neuropsychiatric conditions like schizophrenia through the drugs, right? Through dopamine receptor blockers. So that was kind of my entree into the psychiatric world of neural circuits. And I have a real interest in some of these subcortical nuclei, and there was major outstanding questions about how many different neuron populations were in these nuclei, and what were different types of molecular variation in those populations. And with our DropSeq data, we just kind of solve those very, very quickly, to a first approximation. And so some of the surprises I found were just details of novel and cryptic cell types that had been kind of under our nose in these neural circuits for a really long time, and we just didn't have the right molecular markers to distinguish them. So for me, that was fun. But again, I brought my own experience to it, right? And the fun thing is with like the analysis portals like DropViz, if you logged on and you looked at your favorite genes, right, you'd start to find interesting stories and testable hypotheses that could inspire like more experiments.
Yeah, and I know your project is it's particularly focused on striatal neurons,
Yep.
right? And so can you say a little bit about the striatum and why it's important for schizophrenia?
Absolutely, yeah. The striatum, so many antipsychotic drugs are dopamine receptor blockers, and the striatum receives tremendous amounts of dopaminergic input from other subcortical regions. And so it seems like, at least from the level of drug-based therapy, an important node to mediate some of the symptoms that those living with schizophrenia have to deal with. So also, another kind of interesting angle of the single-cell genomics work, you can start to ask questions about, given that we know now an emerging picture of which parts of the genome tend to be turned on, or which parts of the genome are most associated with psychiatric disorders, we can ask which cell types in the brain use those parts of the genome most, right? And it turns out when you do those type of analyses, the striatal principal neurons are also kind of one of the most kind of hotly implicated based on the genes that they use.
And I did just want to ask you about, I know your project is using mice that have mutations related to glutamatergic genes. So could you say a little bit about glutamate and the role of glutamate in schizophrenia?
Sure. Glutamate is a ubiquitous neurotransmitter in the mammalian brain. And it actually goes back to the genomic variation conversation that we kind of started with. So one way to identify the most important molecules associated with a condition is with human genetics. And there's several different types of human genetic analyses. Some look at variants of genes that are floating around and are quite common in our populations. And others focus on new mutations that crop up in individual people. And those new mutations are especially powerful and impactful to study because they can have a real outstanding impact on your predisposition to certain psychiatric disorders. And it turns out just in the last year or two, the sample sizes of studies involving detecting these de novo mutations, the new mutations, have really implicated the glutamate receptor subunits and families in the pathophysiology of schizophrenia in a causal way. And so my application identifies 2 of these subunits that encode pores on the kind of listening side of the synapse that are activated by glutamate. And it asks if we generate mice that carry equivalent mutations that reduce the function of these 2 proteins, how does that change the wiring of the brain? And so we're going to, the support from OneMind is really helping us address the neural wiring differences in these mutations by helping us continue to develop the SBARRO system for mapping synaptic connectivity. And specifically what we're going to do is in the original SBARRO that I developed, we had to take the system apart. We had to, we could grow synaptic networks in a dish in an incubator, but to sample the RNAs, we have to tease all the cells into like a soup of single cells and then sample their RNAs. And support from OneMind is helping us use imaging-based strategies to track those RNAs in the intact tissue. So we'll use a microscope rather than a DNA sequencer for our experiments.
Well, Arpi, I just want to thank you for spending the time to chat with us about your research. It's really phenomenal, and I think you're just doing such exciting work. So thank you again.
Thank you so much, Carrie. I really appreciate it. Yeah.